Hello everyone, and welcome back to Behind the Bench. So today we're going to go over a relatively recent advancement in genetic engineering, and that is CRISPR. So CRISPR, developed for gene editing in 2013, is a system modeled after a bacterial defense, and it was fairly revolutionary because it's an easy and efficient way to modify genomes in vivo, that's in living organisms, more so than other techniques that are available. So in bacteria, this CRISPR system involves an enzyme called Cas9 cutting invading viral DNA. So the bacteria will synthesize two strands of RNA that help guide the Cas9 enzyme to the viral target. And then it's an endonuclease, so it'll cut within the DNA strand and render it useless. To ensure that the Cas9 does not cut its own DNA, certain sites called PAM sites are required. These are not found in bacteria, so the Cas9 won't bind to bacterial DNA. And we take advantage of these different aspects of the system to generate our own modifications in target DNA. So you see here a double-stranded DNA sequence, and the red sites are these PAM sites that I mentioned. They're required for Cas9 to bind, as you see here. So how this works is an RNA strand called a guide RNA. You'll see it as either gRNA or sgRNA. Will be complementary to this target sequence that we have denoted as black and dark green nucleotides. So the guide RNA will bind to this sequence, and then another portion of this RNA, called the scaffold portion, is bound to the Cas9 enzyme, and in this way it carries the Cas9 over to the appropriate cut site. So you design this guide RNA to target a specific sequence. It will carry the Cas9 over, and as long as there are PAM sites, it will bind and make its cut. So in the target sequence, you'll notice there are two colors there. This is to denote what's called the seed sequence. So the first 12-ish nucleotides of the target are called the seed, and these must be essentially a 100% match to the guide. In the black portion, there can be one or two mismatches, and it's not a big deal. The guide will still bind to it. But in the seed, it has to be 100% complementary to the RNA. And this is something you account for when you're designing your guide RNA. So what will happen is once the guide binds to its complementary sequence, scaffold portion carries the Cas9 over. There are PAM sites, so the Cas9 binds. It will then make a double-stranded break about 3 to 4 nucleotides upstream of the PAM site, and that's where you see this black dotted line. And then we have to look at how the cell will repair this break, and that's where we can cause modifications. So there are two types of repair that the cell will use to fill in this double-stranded break. The first is NHEJ, which is non-homologous end joining. So what this basically means is a ligase will be used to ligate the two ends together, and that's it. This is the most commonly used repair method by the cell, and is not very reliable. It will frequently result in either random insertions or random deletions. These will be shown here in green and red respectively, green for insertion, red for deletion. So there's a complex of enzymes that associate, you have polymerases, and these will be used to create these random insertions and deletions. And HEJ can be used to put in an intentional insertion. If you include a segment of DNA, it might try and attach it to the blunt ends, but that's not as likely. If you want to include an insertion, you typically use the other repair method, HDR, homology-driven repair. What this does is it uses a template that you provide and design as a guide to fill in the breaks. So just like in DNA replication, how a template is used to create a new DNA strand. This template will be used to fill in the break, and through that you can add large insertions such as reporter genes or mutations if you want to create a particular mutation in your gene. So in the diagram here, the top strands are the two strands from the original genome, and then the middle is the donor, and then the bottom shows it with the insertion. So I'd like to thank you very much for watching our video. If you liked it, please give it a like. If you want more content like this, then subscribe to our channel. And if you have any questions or comments about the content or the video itself, then please leave a comment. And thank you for watching. We'll see you next time.